following video demonstrates that the theory of evolution is useful for making sense out of biological observations. For many years, biologists have known that the genomes of eukaryotes, organisms whose DNA is contained within a nucleus, are generally much larger than the genomes of prokaryotes. At first glance, this seems reasonable. More complex organisms might be expected to require more genetic information. However, this pattern quickly breaks down when we compare different eukaryotes. It turns out that genome size does not directly relate to organismal complexity. For example, axolotl salamanders have genomes that contain more than 10 times as much DNA as the human genome. Yet axolotls are obviously not 10 times more complex than humans. This apparent contradiction between genome size and overall complexity became known as the genome size paradox. The resolution of this paradox came when scientists discovered something unexpected. A large fraction of eukaryotic DNA does not code for proteins. Much of the size difference between genomes, such as the difference between humans and salamanders, is not due to a greater number of genes, but to a greater amount of so-called non-coding DNA. Some non-coding DNA exists as long stretches of nucleotides located between genes, but an important portion of non-coding DNA actually occurs within genes themselves. Many eukaryotic genes are what scientists call split genes. In these genes, the coding segments, called exons, are interrupted by non-coding sequences known as introns. Introns are removed by a process called RNA splicing before translation. When a gene is expressed, the entire sequence is first transcribed into an RNA molecule inside the nucleus. But before that RNA can be used to produce a protein, the introns, shown here in green, are removed, and the exons, shown here in yellow, are joined together through the process of RNA splicing. The final RNA molecule, called messenger RNA, or mRNA, contains only the exon sequences. The mRNA then leaves the nucleus, where it is translated into a protein by ribosomes in the cytoplasm. This surprising structure was discovered in 1977 by molecular biologists Philip Sharp and Richard Roberts while studying adenovirus genes expressed in human cells. The researchers were attempting to match viral mRNA molecules to the viral DNA sequences from which they were transcribed. Instead of finding a continuous match, they discovered that the mRNA corresponded to several separate regions of the DNA sequence, with large segments missing in between. Those missing segments were the introns. For this discovery, Sharp and Roberts were awarded the 1993 Nobel Prize in Physiology or Medicine. Subsequent research has shown that many genes in multicellular eukaryotes contain introns, with vertebrate genes averaging roughly 8 to 10 introns each. In many cases, the majority of the transcribed sequence of a gene consists of introns that are removed before the final mRNA is produced. So, what does this discovery have to do with evolution? The discovery of introns allowed scientists to make a very specific prediction about how sequences of DNA should evolve over time. The basic principle of evolutionary biology is that mutations in DNA are the ultimate source of heritable variation. Some mutations affect the structure or function of a protein. These mutations are often harmful and tend to be removed from populations by natural selection. Other mutations have no effect on an organism's survival or reproduction. These mutations are considered selectively neutral and can accumulate over time. Because introns are removed by RNA splicing, most mutations that occur within intron sequences do not affect the structure of the protein produced by the gene. As a result, most mutations that occur within introns are selectively neutral, although some intron regions do have a regulatory function. Here is the prediction. If natural selection acts to preserve functional protein sequences across generations, then mutations occurring in exons are more likely to be eliminated. Mutations occurring in introns, however, should accumulate more freely. This means that when we compare the same gene in two related species, exon sequences should be more similar than intron sequences. And this is exactly what researchers observe. Consider the gene that encodes preproinsulin, the precursor molecule that is processed to produce the hormone insulin. Humans and chimpanzees share a very close evolutionary relationship, so it is not surprising that their pre-proinsulin proteins differ by only 2 amino acids out of 110, a similarity of 98%. The DNA sequences of the insulin gene are slightly less similar overall, but most of the differences occur in the gene's introns. The exon sequences remain highly conserved because mutations in these regions are more likely to disrupt protein function and be removed by natural selection. When more distantly related organisms are compared, for example humans and pigs, the proteins are less similar, and the differences in DNA sequences increase, particularly within the introns. Across many species, these patterns form a nested hierarchy of similarities that matches evolutionary relationships inferred from separate lines of evidence. Modern genome sequencing has allowed scientists to test these predictions on an enormous scale. For example, comparisons of the human and mouse genomes show that protein-coding exons are highly conserved between the two species, even though the surrounding introns often differ dramatically in both length and sequence. Another example comes from comparisons among the genomes of humans, chimpanzees, and gorillas. Across thousands of genes, researchers consistently observe the same pattern. Coding regions change slowly, while intronic regions accumulate mutations more rapidly. Modern genomics provides even more precise ways to measure how natural selection shapes genes. Scientists compare the rate of mutations that cause amino acid changes in proteins, called non-synonymous mutations, to mutations that do not alter the amino acid sequence, known as synonymous mutations. This comparison produces a value known as the dn over ds ratio. When natural selection is preserving protein function, mutations that alter amino acids are removed more frequently than those that do not, producing a dn over ds ratio less than 1. Across thousands of genes in many species, researchers consistently observe this pattern, providing quantitative evidence that natural selection constrains protein coding regions of the genome. Large-scale genomic comparisons also reveal other striking patterns predicted by evolution. For example, many distantly related eukaryotes share introns in exactly the same positions within homologous genes, indicating that these introns were already present in their common ancestors. At the same time, genomes contain rare stretches of DNA known as ultra-conserved elements, regions that have remained almost completely unchanged across vast spans of evolutionary time, demonstrating that natural selection can preserve critical sequences with extraordinary precision. But we can test this idea even further. Consider pseudogenes. Pseudogenes are non-functional copies of genes, essentially genetic fossils left behind by evolution. Because pseudogenes no longer produce functional proteins, mutations that occur within them are not affected by natural selection. This leads to another prediction. If all mutations in a pseudogene are selectively neutral, then they should accumulate at similar rates in both exons and introns. As we saw in an earlier video, the primate gulo vitamin C pseudogene carries the same inactivating mutations across related species. Once a gene loses its function, studies show that the ratio of amino acid sequence changing to silent mutations, the dn over ds ratio, approaches 1, exactly what evolution predicts. This has been documented in comparative studies of vitamin C gene loss and in broader analyses. When pseudogenes from related species are compared, the difference between exon and intron mutation rates largely disappears. So, there you have it. What began as a surprising discovery about the structure of genes ultimately provided scientists with a powerful opportunity to test predictions derived from evolutionary theory. Over and over again, the evidence collected has matched those predictions. Exons remain highly conserved because natural selection protects functional protein sequences. Introns accumulate mutations more freely because most changes within them are selectively neutral. And pseudogenes, where selection no longer operates, lose this distinction entirely. Taken together, these observations form a coherent body of evidence showing how mutation, natural selection, and common ancestry shape the structure of modern genomes.
These observations only make sense using the real science of evolution. I'm Jeremy Moen. Thanks for watching my video.